Greetings everyone, myself Kamalakannan Karupaiah, and the topic of our study is multi-channel auditory cortical responses and source analysis in misophonia, a neurophysiological investigation. So this study work was conducted at All India Institute of Speech and Hearing, Mysore. So background. So misophonia is a condition where individual shows intense emotional and physiological responses to specific everyday sounds, such as chewing or swallowing, ticking clocks. So neuroimaging studies have shown that misophonia associated with structural and functional brain changes, particularly regions such as auditory cortex, amygdala, and insula are involved. Beyond neuroimaging, electrophysiological studies in misophonia have shown inconsistent findings. That is, some report differences in peak latency, while some report differences in amplitude. So this inconsistency highlights the need for a more detailed and systematic investigations. So in this study, we used a multi-channel EEG to capture both temporal and spatial aspects of auditory processing. So we hypothesized that individuals with misophonia would show atypical early auditory processing, and if identified, this could be a potential neurophysiological biomarkers. So multi-channel cortical auditory evoked potential with topographical pattern and source analysis provides high temporal resolution, and it will give insights into subtle neural differences. So aim of the study. Hence, this study investigate auditory late latency responses in normal hearing individuals with and without misophonia. So these were the objective. We have compared scalp topography and source analysis between the two groups. In addition to this, we compared ALLR amplitude and latency parameters at midlane electrodes, that is FZ, CZ, and PZ between the two groups. So moving on to methods. So standard group comparison study design was followed. So written informed consent was obtained from all the participants. Purposive sampling was used, and all evaluation were done in acoustically and electrically shielded room. So participants. So individuals were divided into two groups. One group is with misophonia and the other group is without misophonia. A total of 40 participants were recruited. All participants were in the age range of between 18 and 30 years. These were the mean and ST of age range. So individuals with misophonia were recruited based on the criteria proposed by Schroeder et al. And MesoQuest questionnaire. In addition to this, severity of misophonia was administered using revised Amsterdam misophonia scale. So these were the evaluation done in this study. Our major focus would be on ALLR. So procedure. So stimulus presentation system. So STIM2 software was used to present the stimulus. So EEG recording system, 32 channel neuroscan equipment was used. Curry 9 software was used to do acquisition of EEG. So EEG analysis were done using BISA research and BISA statistics software. So this was the 32 channel layout used in this study. In addition to this 32, we have used vertical electroocculogram and horizontal electroocculogram electrodes to monitor the eye movements. So stimulus used was DA syllable with a duration of 130 milliseconds and an inter-trial interval of 2 seconds. So moving on to results. The Shapiro-Wilk test was carried out to see whether the data was normally distributed or not. Here the data was normally distributed, hence we proceeded with independent t-test to compare the group between the groups. So this figure represents the mean SD and individual data points of each participant. So green circle represents control, whereas orange represents misophonia. So from this figure, it is interpreted that individuals with misophonia exhibited early latencies of P1, N1 and P2 at midlane electrodes, that is FZ, CZ and PZ. So as seen in the figure, there is a difference in the latency of all the peaks. It is highlighted here. So amplitude. So here in amplitude, individual with misophonia showed reduction in only in N1 amplitude, as highlighted here, at all the three electrodes, that is FZ, CZ, and PZ. There was a reduction in only N1 amplitude. There was no differences in P1 and P2 components. This figure A represents butterfly plot grand average waveform for control group and misophonia group. So from this waveform, it is seen that individuals with misophonia exhibited earlier latencies of P1, N1, and P2, and there is a reduction of N1 amplitude. And scalp topography reveals individuals with misophonia showed central to parietal distribution, and control group exhibited frontal to central distribution. At these four electrodes, there was a differences in scalp topographical pattern. So in addition to topographical analysis, we did source analysis. So source analysis indicated in misophonia group, there is a prominent activation in medial frontal gyrus, whereas in control group, exhibited greater activation in left temporal lobe. So to conclude, individual with misophonia shows earlier P1, N1, P2 latencies and reduced N1 amplitude. So this suggested there is a altered in early auditory processing, and it also highlights the altered temporal dynamics and spatial organization of auditory cortical responses in misophonia. So what next? So future studies. So longitudinal studies with trigger specific stimuli can be done. Advanced analysis such as time frequency and functional connectivity analysis can also be done, and treatment based studies can also be done. This is one of our publication from our research lab. So we have given the link here. So please go through. So acknowledgement. I would like to thank the organizers, TRA Academy, for this opportunity, and all the participants in the studies, and Dr. M. Pushpavati, Madam, Director AISH, Dr. Ajit Kumar U, Sir, Professor Audiology, Dr. Neeraj Kumar Singh, Sir, HOD Audiology, Center for Hearing Sciences Lab, for the space and the equipment provided. And I would like to thank my dear friend Mr. Rakesh for his valuable inputs. Thank you for your attention.